Most of today is actually going to be you guys working and grouping and reporting out. But what I wanted to start with was this question up here, which the note said, do while waiting for class. Nobody did it, and I didn't assume anybody would, but that's okay. We can at least talk about it. So it says, you infect cells in culture with a virus. What might the one-step growth curve look like when? You add an antibody that binds to the receptor that the virus attaches to as well. How would a one-step growth curve deviate from this, or would it deviate when you have an antibody that binds to the receptor? So in other words, you have two conditions. One where you have cells and virus, one where you have cells, antibody, and virus all at one time. How will this change? You're thinking the virus wouldn't be able to enter the cell? So in other words, we'd see maybe something like that, just a flat line. You could argue that. And I don't know what the right answer is, okay? I don't think there is one right answer to this. You might see something in between these. Oh, damn it. Now we need another vote. Jonah, which of those 4 bacteria do we need to create phage for? Nobody should sway the vote. Okay, it's still Pseudomonas fluorescens. So that's absolutely one option, right? If we had an abundance of antibody, if it wasn't limiting, probably this is what we would observe. No infection would take place. It's more likely, given another possibility would be something in between the two of these, where we might see most of the virus being inhibited, maybe some actually getting into the cells, so maybe not as much as of a dip in the eclipse phase, and probably not as much of an explosion of virus either, because not every single cell is being infected. Okay. Either would be fine. Goal here is the justification piece. We know that receptor is required for entry. It's a prerequisite it's the first step of the infection, and so if you block that, you block all the subsequent steps as well. What if the virus is a naked virus and clathrin-mediated processes are blocked? So you have a drug, for example, that blocks clathrin from forming its nice three-dimensional structure. Grace? Okay, good. So the follow-up question, do naked viruses require endocytosis to enter?
Yes.
It does. You're absolutely right. So in that case, we might see what? Well, we might still see attachment, right? There are a couple of possibilities here. Again, flat line. No change whatsoever. Because the attachment step actually can be reversible. Okay? The other possibility is we might see attachment, a decrease, and then that level just stays low as the virus is being absorbed or attaching to the host cells. But because there's never any point of entry, you might still have viral particles present there. How about this last one? Virus is enveloped, enters by pH-independent methods. Clathrin-mediated processes are blocked. If it's pH-independent, sorry, what was that?
Ideal situation.
What do you mean ideal?
If the virus is enveloped, it would do what's it called, the fusion.
If it's pH independent, it would do fusion. Good.
Enters by pH independent, and then the chlorophyll is just for the endocytosis.
Exactly. The clathrin's only required for the endocytosis. So in terms of the virus, yeah, this is a great thing, right? It's going to have no effect whatsoever. We know that the pH-independent methods, as Michaela is pointing out, good terminology here with the event is going to be a fusion event at the cell membrane, it doesn't require the virus to enter through an endosome. And so in that case, we'd see no effect whatsoever. The clathrin inhibitor would not do anything to block replication of this virus, or at least that's what we could assume. Okay. Be practicing these types of problems. Bless you. This is this was a review one that I have kind of on the list of ones for this particular section or chapter that we're looking at. So be practicing these. These are, again, example exam questions. Okay. All right. Let me see what we're doing next. I can't remember anymore. Oh, okay. We're going to cover the last two steps of the life cycle, and then move into something a little bit, well, I'm going to have you guys actually doing some work for a while. The last two steps that we haven't covered yet, remember what we have covered: absorption or attachment, entry/penetration, biosynthesis, assembly, and now we're on to the last two, maturation and egress or exiting. Maturation is the point at which we find that viral proteins are becoming fully mature. What we mean by this is, for example, some of the glycoproteins that make up spikes or the receptor binding proteins, they may require sugar molecules to be put onto them. That's part of the maturation. Any final cleavage of polyproteins is another example of a maturation process. At this point, this is the point at which a virus becomes fully active, or a viral particle becomes infectious again. And this can occur in the host cell, it can occur outside of the host cell. For some of those envelope viruses that exit by the opposite of fusion, which is budding, the maturation step doesn't actually occur until that bud has come off. And we'll look at that in the next step. So again, maturation this is kind of a color of gray here. This isn't necessarily a distinct step. It could happen at the same time as assembly. It could happen at the same time as exit as well. And the egress is that exit. So let's take a look at a couple of mechanisms here. Here are a couple of different images. The one on the left is an example of lysis. So in that particular image, we have a bacterial cell. You can see the remnants of the cell membrane and cell wall here. And all of these little circular-like structures are phage heads. Okay, so we've got something on the order of about 100 phage exploding from a single bacterial cell. Woohoo! All right. On the right here is a different mechanism. Okay. Lysis we're going to find is common with naked viruses. Not all naked viruses exit by this mechanism, but some do. Some will actually exit by exocytosis, the opposite of endocytosis. This here is a little bit different. So this image here, I can't remember which virus this is showing, but you can see different steps, different numbers here representing different steps in the process of this exit. So 1, you've got the formation of a little bud here. 2 becomes kind of polypy looking. 3, at this point, you're starting to close off or pinch off the membrane between the envelope and the cell membrane, and then 4, full release or exit of the viral particle. This would be common for what kind of viruses? Envelope viruses. Not all enveloped exit by this strategy. Some do. This is what we call budding. HIV is an example of such a virus that exits by budding. And in the case of HIV, what this image is trying to show you is that what we have are these rafts or locations where the viral proteins all congregate and we start to get self-assembly at the membrane structure. So here we have glycoproteins At this point, this GP 120 and 41 are still a single protein. It has to undergo a final cleavage step, which occurs after the bud has broken off. We have protein known as P55, which serves as a scaffold to recruit the RNA and reverse transcriptase. We have polymerase precursors that are going to assemble here as well. So we're starting to get those molecular interactions that are driving the self-assembly of this protein right at the cell membrane. And as that virus is kind of pushing outward, then what's going to happen is this will close up, and we've got our fully formed virus at this point. The other way by which some of these viruses, both enveloped and naked viruses, they both will use exocytosis as a mechanism. So they may go through the endoplasmic reticulum, through the trans-Golgi network as a way to undergo their final maturation and then ultimately exit from the host cell. Okay, so those are all the steps. All right, so let me go back to the very first slide real quick. So a couple of things here. List the key steps, describe what's happening. That's what we've kind of just gone through. All right, we've gone through some details. We'll get more into these details as we talk about specific types of viruses. But keep in mind the key features. We've correlated the steps of this with the one-step growth curve. Now we're going to move on to biosynthesis again. So what I want to do today is have you guys tell me how biosynthesis occurs. I'm going to give each group a single virus. What you're going to see on here is a viral name, or a virus name. So this one is Maloney-Murine leukemia virus. You'll also see the family. So, for example, Maloney-Murine leukemia virus is a retroviridae. That should tell you something about it already. You guys have Synvis virus, which is a togaviridae. Okay, they like to party. We've got rotavirus, which is a rheoviridae, and then we have a couple of others. I guess I'll cover these too. But here's what I want you to do. So I'm going to do an example of, which one was I going to do? And what do I want to write with? Okay. Oh, that's right. I was going to do variola. So variola virus, member of the poxviridae, so related to smallpox. The reason why I'm giving you the specific, or the family, is to point out here that there are commonalities. You're looking at a specific virus, but what you're going to tell me about this one virus is true for others in the same family or class. So there are 5 questions on here. First one is, what type of genome is it and the size of this genome? So variola virus has a genome of about, if I'm not mistaken, it's about 200 kb. I'll double check that to be certain. It's double-stranded DNA. Okay. Next question is, excuse me, what are the mechanisms to encode for multiple proteins? So for example, we've talked about polyproteins. We've talked about leaky scanning, frame shift, or ribosome skippage. In variola, they actually have a separate promoter for each mRNA. So in other words, they produce monocistronic mRNAs, each with its own promoter. These have no introns whatsoever. And not that that's important per se, but that actually should tell you a little bit about this virus. Next question, polymerase for replication and transcription. You tell me. This one's a DNA virus. What does it use for replication? DNA-dependent DNA polymerase. And transcription? DNA-dependent RNA polymerase. The events of replication. This is you telling me basically how this occurs. So this virus, and this does not need to be in great detail, but for some of the viruses like plus single-strand, you can't have a plus single-stranded RNA virus and just make more plus single-stranded RNA. There has to be an intermediate. So what is that process? For this, it's pretty easy. Replication starts from an origin, and it moves from that origin of replication. The DNA polymerase moves in both directions to make copies of that template DNA. This virus replicates in the cytoplasm. That tells us something about it as well. What does that tell us? Has to have its own, exactly. So this one has to have its own DNA-dependent DNA polymerase. And then the second part are the events of transcription. And in this one, pretty darn simple. For transcription to occur, it uses the DNA-dependent RNA polymerase, begins transcription from individual promoters, and drives transcription of the RNA here from that DNA template. Okay. Let's go for about 10 minutes and see where we get with this. Okay, if it takes more time than today to finish all this up, that's fine, because what I want you guys to do is to kind of work through this relatively quickly. The goal is not to get into too much detail, but to focus on the major points. And then we're going to report out. I put whiteboards on here so that you can feel free to write all of your responses right on there. And I'll go grab my phone so that I can take pictures of each of them and we can post them afterward today too. So you don't have to feel like you just start taking notes the whole time. All right, so use your resources. That's the last one. Slides have some information. The other useful source here, Viral Zone. So if you type in the name of the virus or the name of the family, that should actually take you, eventually you should be able to find your way to a site that has some of the basic information about the viral genome. All right? All right, go ahead.